So now I'm going to do a video regarding biochemistry. I'm going to do an overview, so this is brief. Alright, so we have three things I want to talk about first. I'm going to talk about insulin. So insulin, okay, is secreted by the pancreatic beta cells. Glucagon is secreted by the pancreatic alpha cells. And somatostatin is secreted by the delta cells. Somatostatin is the one that regulates glucagon and insulin, and it's the main regulator of those two. Then we have insulin. So what insulin does is decreases glucose, and it does this by using phosphoninositol-3 kinase (PKB) and acting as a phosphatase. So it removes phosphates, and it's important for anabolism. So when I'm talking about anabolism, I'm talking about creating molecules, like for example glycogen, creating amino acids, fatty acid synthesis, and also energy, like doing the citric acid cycle, glycolysis, among others. Okay, glucagon responds when glucose is gone, and it's important for catabolism, meaning breakdown of things, breakdown of glycogen, breakdown of amino acids, breakdown of fatty acids, also called fatty beta oxidation. It also involves synthesis of ketone bodies. It reduces glycolysis, it reduces citric acid cycle because it just needs to increase glucose. It induces gluconeogenesis, so it's part of catabolism. It's the opposite of insulin. Glucagon does not want to build molecules up, it wants to break things down. And it does this using G protein coupled receptors. The alpha subunit detaches from the gamma and the beta subunits of the G protein, and then the alpha subunit is the business end. It activates adenyl cyclase, and adenyl cyclase it's gonna increase cyclic AMP levels. And when cyclic AMP levels go up, pKa goes up as well, and it functions by adding phosphate into molecules. Okay, the next thing we want to talk about are the specifics of the tissues. So adipose tissue has no glycogen. It mainly functions to store triacylglycerols, okay? And when we have, for example, glucagon levels that are going up, we need to break the triacylglycerols down into glycerol and fatty acids. The glycerol is going to get sent to the liver, and the liver is going to utilize the glycerol to either to make amino acids or to get energy. Okay, the muscle is very selfish. It utilizes amino acids, glycogen. It has glycogen, and it does gluconeogenesis and all of that. But for gluconeogenesis, it only uses glucose for itself, unlike the liver. The liver is like the lover. It creates ketone bodies, and it uses a lot of energy resources to feed the brain. And it's not selfish like the muscle. It has everything. It has glycerol. It has glycogen storage. It can do gluconeogenesis. It can do ketogenesis, among other things. Now the brain primarily does glycolysis, so its focus is on metabolizing glucose. It usually likes glucose even during times of starvation, but when we run out of glucose, because for example we utilize all the glycogen and we can no longer do gluconeogenesis, and we already utilized all the fatty acids and fatty acid beta oxidation, then that's when we get ketone bodies. So the brain has to rely on ketone bodies instead of glucose. Okay, so for glycolysis, it's really simple. We go from glucose to glucose 6-phosphate, from glucose 6-phosphate to fructose 6-phosphate, then to fructose 1,6-biphosphate, then to glyceraldehyde 3-phosphate or dihydroxyacetone phosphate that eventually gets turned back into G3P. Then we get glyceraldehyde 1,3-biphosphate, then we get glycerate, and then we get phosphoenolpyruvate and then pyruvate. So it's a long chain reaction. The rate limiting step is actually phosphofructokinase 1. Phosphofructokinase 1 is responsible for transforming fructose 6-phosphate into fructose 1,6-biphosphate. Okay, we also have phosphofructokinase 2, And this one, it's responsible for making fructose 2,6 biphosphate, and fructose 2,6 biphosphate activates phosphofructokinase 1. Okay, so that you can go this way. Otherwise, you go backwards. All right. Another important mention that I have to state is that hexokinase is in the muscle. So it takes glucose to glucose 6-phosphate. Hexokinase is in the muscle, and glucokinase works in the liver. How do we remember this? Because glucokinase has an L and liver has an L. Okay. We also know that glycerol 3-phosphate can also be shunted back into the PPP system. Okay. And this system basically creates NADPH and ribose. Now, what is NADPH? NADPH is involved in reductive biosynthesis. Okay, so it's creating molecules, usually like for example fatty acid synthesis, by reducing them, which is the opposite of oxidation. And ribose is also made, and ribose is important for DNA synthesis. Okay, so now we go from phosphoenolpyruvate to pyruvate, and this is done through pyruvate kinase. Pyruvate kinase is actually activated by phosphofructokinase 1. Okay, so it activates pyruvate kinase. And this whole reaction generates two